I want to make now here just 3 more points about this discussion of origins. The naturalist atheist starts from the point that they say that there is absolutely no design in nature. They say that there is not any design in the constants of nature. There's not any design on this planet. There's no design in biochemistry, and there's no design in biology. But all has been created by the neo-Darwinian mechanism, which has been extrapolated to biochemistry and the cosmic origin. This is a very interesting claim, and I want to look at it critically. After this presentation, we will see the film about the constants of nature and physics. And it is the most interesting film in this area which I have seen myself, The Privileged Planet. There's many scientists explaining why we can talk about design in the constants of nature, in physics, and on this planet. And as far as the design is concerned, we went over the luxury design, we went over the extra design in man. And now we look at some specific examples of design which are very interesting in this discussion. There was a communist scientist, an atheist scientist named J.B.S. Haldane, who said in the year 1949, 2 years after I was born, but I don't remember it from there. But he said, this atheist and communist famous scientist J.B.S. Haldane, he said that if we ever find a wheel, a round wheel in nature, or a magnet in nature, then he must become a believer in God. Unfortunately, Haldane died before we started to find machines in the nature. Now we have lots of machines in the nature. And had Haldane lived to see this, I don't know if he would have become a Christian. Maybe, maybe not. But Anthony Flew became a deist, a believer in the Creator, after he saw the evidence. So what I want to do is just to take an example of design in nature. Because logically thinking, let's say you go to a junkyard and you find an electrical motor. What you think first is probably not that this electric motor in the junkyard has assembled itself there by chance. Logically and intuitively, your first thought is that an engineer, an electrician, has assembled this motor. That is quite logical. So you can draw the conclusion of a designer when you find an old electric motor in a junkyard. Now, when we find the motors in the nature, can we draw the same conclusion or not? Here's the bacterial motor. This is well known. The bacteria like E. coli have fine motors like this, and E. coli bacteria typically may have 6 motors like this. They're turning and they're helping the E. coli bacterium to go to where it wants to go. Bacterial motor. This has been studied very well. We know the details. We know there's about 40 components, parts in this motor. We know the genes which code for the motor and how they function. This is the same thing in Finnish. This we published with Professor Matti Leisola, who is a professor of bioprocess technology at the Technical University of Espoo, Finland. He has been the dean of the Technical University of Espoo. He has been a Christian all his life, and he has fearlessly defended design in nature. He's a very good scientist. He is both docent in Zurich, speaks German, and in Helsinki, and he's a professor and the dean of the faculty. Now he's on pension. I've been working together with Professor Leisola for 25 years, and we just a month ago gave out this book, Evoluutio ja biologinen informaatio. It's based on German scientists' original work. I know many scientists in Germany, like a biology professor Siegfried Scherer in Munich University, who has been helping us. It's a funny thing how once you say that you believe in a designer, they start to throw mud at you and talk nasty things about you. I can tell that Professor Leisola was employed by the Finnish sugar company when he was younger. He went into the Finnish sugar company, and he was known as a Christian, and the research chief at the Finnish sugar company said to Professor Leisola, he was not professor then but a docent, said to him that, "You now, shut up here in this company, don't talk about Christianity or we'll kick you out. You will be not surviving long in this company if you start talking about Christianity." Well, Leisola continued to talk about Christianity, and it took 2 weeks. And the research chief was kicked out, and Leisola was made the new research chief. Because he's so good, he's good scientist. Well, these things happen, but he's fearless. Well, of course, he said that he was very much afraid, but still he has been systematic in defending creation. So we know a lot about the details of the motor. Here's a propeller. And one thing that strikes you when you look at these biomolecules is that they are ingeniously designed. And another thing, they are beautiful. They are really beautiful also. There's a beauty aspect and the design aspect. These flagellin proteins, to make the propeller for the motor of the Colibacterium, it needs 20,000 molecule systems like this, and they come through this hole and add up and build up the propeller. 20,000 units. It's really nice. But what would be a motor without a clutch? You know, the car has a clutch. I have an old-fashioned car with a clutch. I like it because my legs move when I drive, so I don't get the thrombosis. So the clutch is good. But this motor has a clutch, like a car. It actually has a clutch. And the more we study, the more we see. This molecular clutch is thought to dock on the rotor, and so on. And this bacteria must know where it's going, of course. It has a nose. It can smell food. It can smell poison. And it has a nose, and it tells the motor where to go. These E. coli bacteria have a chemosensory and signal transduction machinery. It must compute the information. There is food, let's go there. There is poison, let's go away from there. And it's interesting that food and poison and also sunshine, if there's not enough ozone layer, so the bacterium sees that this is dangerous for me here, too much ultraviolet B, I go away. It can sense food, poison, the length of the electromagnetic radiation. But that's not all. Did you know that if I make a hole here 2 kilometers deep, right here, what do I find? Do I find bacteria? Yes, I find bacteria. There are bacteria. How do those bacteria know where to go at the depth of 2 kilometers? They have magnets. And this is where Haldane comes into picture. Haldane said, if you find motors and magnets in nature, I will start to believe in God. So these bacteria produce magnetite crystals, very nice magnetite crystals. They are so fine that we cannot produce such perfect crystals. Therefore, in the industry, these bacteria are used to produce these crystals, which are perfect. But the crystal is too short. They put the crystals like wagons on a train, a long train, and then they get a needle which is sensitive to the strength and direction of Earth's magnetic field. So they have a navigation system, they know where they're going. Very smart. They sense it, they relay, compute the information, they relay the information to the motors. So bacteria are extremely complicated, as you will see. It's not a simple cell at all, as you will see soon. They are very intelligent. Actually, this should be a planet of bacteria, because bacteria can survive at 122 plus centigrade in hot springs, 2 kilometer depth, extreme pressure. They are in the air 10 kilometers high. They are inside ice. All over the place you have bacteria. Why should there be men? Because bacteria have children every 20 minutes. We have children if we succeed. It takes 9 months. And why should evolution be so stupid as to create like men, which have so much problems? Bacteria can make it all over the place. This was funny because Stephen Jay Gould, who's an atheist, he said that this is a planet of bacteria and we have a problem explaining why there is something else than bacteria. That is a problem. Actually, one of the trickiest questions in evolution is why we have a man and woman. Because for the ladies here, it would be better to produce your children by dividing in two. Because why take this package of chromosomes from some unknown type who has 85% more mutations than you? That's not good. So I recommend to you ladies, divide into two to have kids. This is true. It's very difficult, impossible to explain which selective Darwinian mechanism produced like a man and woman. Where the Lord says that, haven't you read that in the beginning God created man and woman in the beginning? Well, that was a little thing for you ladies. But if you look at the details, animations of bacteria, it's all over the YouTube. Here's another machine, the ATP synthase, which is in our mitochondria, produces the ATP, which helps us survive. This is a rotor engine also. ATP synthase. It works with protons, produces 100 ATP molecules in a second. Very complicated and interesting rotating machine. Well, can intelligent design, like motors, be seen in nature and tested? Yes, we can, said Obama. But we can see that, we can test it. Evolutionists claim that the idea of a designer is based totally on faith, because there is no design in nature, and design in nature cannot be tested. Well, I think there is design in nature, and design can be tested. So this claim is false. that's about the Haldane. Well, these machines have sensory proteins for food, for UV ultraviolet radiation, and they can build magnetite particles, which we went through already. They're very, very complicated systems. So bacteria are not simple or primitive. That's racism to claim that the bacteria is simple or primitive. Because today we know that one bacterium has about 1,000 to 8,000 genes. But a population of E. coli can have 16,000 genes. That is called the E. coli pangenome. It has like they have a bag of information. You take that bag, you take that bag, you take that bag. Together they got all this information, and then the guy says, well, I need this information, please come here and give me that. They give information to each other. This is called the E. coli pangenome. And today it has been estimated, this is quite new knowledge. Today it has been estimated that the E. coli populations probably carry with them up to 45,000 genes. How many genes does a man have?
45.
No, 19,500. It's going down, 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 down. Twice as many genes E. coli population altogether as we do. So lift your hat to the E. coli. Very interesting thing. So all the old concepts, like claiming that bacteria are simple or primitive, they are wrong. They are not true. This just shows how science goes forward. You see here, one has sequenced now 186 different E. coli, and 15,000 genes have been found. And the estimate is that there will be, after this is completed, there will be 45,000 or something like that. So bacteria have messenger proteins. They relay information to the motors. The motors have a clutch and a brake. This is funny that when I was driving here, I saw that the Estonians are very dangerous drivers. Because the speed limit was 70. I drove 70 because I want to respect, I'm afraid of the police. I heard the police is dangerous here. So they tried to find the Finnish and find them. So I was driving at 70, everybody was passing me, even in the trucks and in the cars. But people are very dangerous. But the E. coli doesn't want to go too fast. If it thinks there's probably food there, let's go and check. It doesn't rush. It puts on the clutch, and it's a brake, go a slow speed. Maybe there's nothing there, let's go slow and not waste energy. It has a clutch and it has a brake to adjust the speed. The motors have a clutch and a brake, proteins that steer to the right direction at right speed. Is this motor better than our motors? Yes, it is. Because the E. coli can make 6 new motors in 20 minutes. My car is 15 years old. I'm waiting for the motor to break. But if it was an E. coli motor, I would just have to sit there and wait 20 minutes for the motor to build a new. And the motor is carrying with it 300 reserve parts and system to repair motors on the run. 6 motors, one motor breaks down. It takes from inside the bacterium the reserve part. It doesn't have to go to the motonet or whatever. It doesn't have to go to the service. It is carrying the reserve parts. It knows that it might break down. It anticipates this. You see, this kind of anticipation in biology, if it was just a blind thing, why should all this be there ready? Repair system, spare parts, everything. It's a wonderful machine. It's better than the electric machines we construct because they make copies of themselves, they repair themselves, and they carry spare parts themselves. So it's even better than our motors. 40 different proteins carry out this structure and construction. And there's an instruction book also in the bacterium that contains 60,000 letters for a construction and operation manual of the motors. So the motor speed is 20,000 rounds per minute. And the bacterium is very fast. It's faster than Usain Bolt, because it can make 100 meters in 3.6 seconds. So these are very fast bacteria. But there's even a formula bacterium, because the C bacterium has a V24 motor and its speed is up to 100,000 rounds per minute. And the C bacterium uses energy from natrium ions, sodium ions. E. coli uses protons, C bacterium uses sodium ions, and our motors use electrons, all different things. But look at this V24. They got in Jaguar, I think, they got the V12 engine, which is very expensive to repair. I don't have one like that. I got a V6. But this does better. This C bacterium has a V24 motor with 7 propellers to go really fast. And look at the construction. It's inside a bundle, and yes, the motor's turning in this direction, turning the 7 propellers to go at them extremely fast. So somebody says there's no design here. I think there's lots of design here. So it's just, if you don't want to see design, good for you. But I see design here. V24 motor. This bacterium is like a 0.2 volt battery which turns a super ultra nano electric motor. The gear is changed instantly because the rotor turns only 1/4 of a turn and it can go backwards. The direction has changed. The motors are so small, and on the cut surface of a human hair you can place 8 million motors. It's pretty small stuff. I mean, that's a nanotechnique. The bacteria carry with them hundreds of spare parts for repair. No need to go to spare parts store or service station. And as they divide, they build new motors automatically in 20 minutes. This is a really fine engine. And of course, the Darwinists have always an explanation. Now is the Darwinian explanation. Evolutionists, because they don't see any design here, they claim that the parts of the motor have been collected from parts that the bacterium uses for some other purposes. And they say that the parts came from organs that they use to inject poison into cells. Bacteria can be very nasty, they can inject poison into cells. So what is this claim? There is actually an injection needle in bacteria. It can inject poison into cells. And it's a nano injection. But could this injection machine turn into a motor? No. Maybe 10 parts of the poison injector could be recycled and used in the motor. But the other 30 parts are totally new. Evolutionists claim that this motor evolved from the poison injector is like saying that parts on the shelf of a store can build a motor spontaneously. But we must, however, remember that the motor must be put together in the right order. Everybody who has repaired engines knows this. You cannot put it together just as you wish, in the right order. There must be also that system. For this, a regulation mechanism must exist. And also the nose, the chemosensory, navigation, information, computing systems, evolution must also be explained. If the bacterial nose says there is food, then the information must be developed and given to the motors to go to the right place, of course. It is so complicated, we don't know how the bacterium does it, but it does. The origin of the molecular motor is a question of faith in light of our knowledge today. Maybe someday its evolution will be explained, or maybe it will never be explained. But if you meet somebody who says there is no design in nature, you can take this motor example and ask the atheist, and how do you explain that? And beware, he will explain it by saying that the poison injector evolved into the motor, but you have an answer to that also. So we go into this new book which came out a month ago by me and Professor Leisola. We go deeply, very deeply into the question of the motor. We go very deeply into the question of, for instance, the Aaron staff, the flower, which is at your home. It's funny that I've been working all summer with this, what is the name of the plant in Estonia? Aaronikeppi. I was working with the Aaronikeppi plant for all summer, and I go to their house and they have this Aaronikeppi in the room. But it's a funny flower because it has a cup like this, and when the evening comes, it starts to burn starch to get some warmth and the smell. The insects come in there, they slide, it's slippery, it produces something which is slippery. The insects fall into the cup. They can't come out because it's like a prison. There's hairs which prevent the insects coming out. But the plant senses, now I got some insects. Let us wait till the insects go around inside the flower long enough to get enough pollination on their back. And after the flower looks at its watch, now they've been there for 15 minutes, now I take the hairs away. The flower takes the hairs away and the insects disappear, go to the next flower and pollinate. So this is a funny mechanism, and that is also luxury design, because why not make it simple, just an open flower like this? No, it's a nice extra luxury thing. So the nature is actually full of different mechanisms of pollination. All flowers almost have different ways to pollinate. So if evolution was true, why wouldn't just one way be enough? Well, the evolutionists work also from their own worldview, as they do science without considering the possibility of an intelligent designer. When God is not considered, it is astonishing what scientists are willing to believe. And it's quite obvious that biomolecular motors have been designed. Why don't the scientists see this design? The answer is in the Bible. People love darkness. They want to sin in peace. So they love darkness. In other words, they close their eyes to design and instead invent evolutionary stories. I can recommend this book in English, Stephen C. Meyer's book, Signature in the Cell. It has lots of this kind of information. He's a scientist who has written from the creation point of view. But remember, it is very dangerous to talk against evolution. This can be seen in Ben Stein film, Expelled, No Intelligence Allowed. We talk about this, the biochemist Jim Tour. We talk about these 1,000 scientists who put their name on the internet site Descent from Darwin. They risk their career. A recent lawsuit was this scientist who was fired, after he found soft tissue on a dinosaur fossil. He got fired immediately. Because if you find soft tissue on dinosaur fossil, you're dead. Because that shows that the dinosaur is maybe 2,000 years old. You cannot publish stuff like that. You will be killed. So he got fired. But he found soft sheets of fibrillar bone from a fossil of the horn of the dinosaur Triceratops horridus. This is where you can find more. Just one publication and you're out. This is the Triceratops dinosaur. From this horn he found biomolecules, which is interesting. And here's some more stuff. I don't want to go into the details, but it explains in the scientific publication what the soft tissue was like. You find this kind of buried dinosaurs, which more tells about the catastrophic burial of dinosaurs. Here's the dinosaur, small dinosaurs. Yes, the biggest one was found last week. A supermassive dinosaur was found last week, but it was published last week in Argentina. Weighed 65 tons, 26 meters long. There's a toe bone of this giant dinosaur. There's somebody lying on the femur of the dinosaur. But this bone is extremely important. This is a femur bone of a dinosaur which was found some years ago. This can be 2 meters long. And this was found in Montana in Hell's Creek. And as the name says, it's a very nasty place. And they went there by helicopter because it was difficult to reach on foot. They went there by helicopter. They found this. But this bone was too big to fit into the helicopter. So they broke it in two to get it into helicopter. The researchers said when they broke this bone into two, it didn't smell so good. It smelled rotten. And one bit of the bone in the laboratory fell accidentally into a jar with acid. And the acid dissolved the bone away. Remark, the bone was not turned into stone, but it was bone, rotten bone. What was left after the rotten dinosaur bone in the inside of the bone was dissolved. That is caused the scammer. This. Here are region, they bored later on the bone, found some more. They found fibrous structures that are still present. The meat we just ate, it was not fibrous, it was very good. But sometimes the meat is fibrous, you know, it's But they found this fibrous dinosaur soft tissue. This was found inside this dinosaur femur, which was not fossilized. What does this mean? Well, I think everybody understands that if we put a bone into the ground, put some sand over it, and then come back after 68 million years to see what is left of the bone, nothing. But now they found that the bone on the outside was fossilized, on the inside it was rotten, and there was soft tissue. Now it's very challenging to explain how soft tissue could survive for 68 million years. This points to the fact that maybe this dinosaur was not 68 million years old, but younger. And even more, from this tissue they took a microscope study, and here's the dinosaur blood vessel, and here's a dinosaur red blood cell. How do we know that this is a dinosaur red blood cell? Because it has a nucleus. Our red blood cells do not have a nucleus, but lizards do have. So this was confirmed. This is the real thing. Dinosaur blood sample. How is it possible that this survived 68 million years ago? It's not possible. Of course, this has lived maybe a few thousand years ago. Well, of course, there's always the evolutionist explanation. They say that because the red blood cells contain iron, the iron would form a scaffold to hold these tissues together. But this is not true. Because recently there was found dinosaur skin. They found a dinosaur fossil with something funny on top of it. They said, what is this? They studied more closely. That is dinosaur skin. I mean, if you have shoes which you don't like and you bury them in the backyard, but then they will be again in fashion after 68 million years, you go and dig them out. Is there any shoes left? No, because bacteria, they like to eat the skin. Of course they eat the skin. It's a bacterial food. But now it's been found, this is dinosaur skin. It has been published in a secular journal quite recently. This is true, dinosaur skin. Examples like this, we need just one example like this to destroy the neo-Darwinian myth. We got very many examples like this. Look at the scientific text. This is from the scientific journal. But perhaps the greatest question they are trying to answer is how the fossil remained intact for around 70 million years. That is a great question. This is not all. Recently was discovered 505 million year old chitin in the basal demosponge of this Vauxia gracilentum. What is chitin? Chitin is a derivative of glucose, and its structure is comparable to the polysaccharide cellulose. Do bacteria eat cellulose? Yes, they do. They love cellulose. So how can you think that in the ground you got a 505 million year old chitin which the bacteria didn't have time to eat? I mean, this is getting really worrisome for the neo-Darwinian idea, because you find fossils 500 million years old, 200 million years old, 100 million years old, 50 million years old. They all contain soft tissue. It shows that they are all maybe a couple of thousand years old, and they have been buried in a catastrophic flood. I don't have time to go into this, but I have a set on the flood question. I will talk about that maybe tomorrow at Axi Church. I will talk in Finnish, but it will be translated into Estonian. There's an exhibition on creation opening in the Axi Church about 20 kilometers from here. I will be talking there. Maybe we'll go into the flood, which is an interesting story. So here's chitin, it's a modern insect, and here's the chitin that they found in the 505 million year old fossil. But this is silly. You don't find the 505 million year old food for bacteria. They would have eaten it, of course. But this is proof that there is soft tissue all over the place. How about this? They found a squid with ink. You are so young people, you have probably not written with the ink. When I was in school, we had a steel pen. You would have to put it into the ink and drop something on the paper and get 0 for the exam, and then try to write with the ink. It was not so easy. But with this ink, you can still write. The scientists took the ink from there and wrote the squid's name with the ink. It was fantastic. And in the original text, 155 million years old, amazing scientists. Of course, it's amazing. How can you have the ink sitting there, 155 million years old, just waiting for us to use it? Because fungi destroy ink. It's as simple as that. Squid are my favorite animals. I described in that book about animals how the squid changes color in 1 second. Because the squid is like a KGB agent. It can change its cloak in 1 second. But the squid is very funny because if it finds itself in the bush, it starts to resemble a bush. If it's among stones, in 1 second it looks like stones. But what is more funny, what is my favorite squid, is a squid in Tahiti. It's in Honolulu. And it's a little squid like this, and it goes swimming at night. And the waters are very clear, and the sand is white. And when the squid is swimming there, it projects its shadow on the white sand. And the shark sees there's a shadow, comes and eats the squid. But the creator has found a solution. It has put some lamps here on the stomach of the squid, so when the shadow is there, the squid will light the lamps and take the shadow away. And even more, if the moonlight is strong, then more lamps must be lit. But when the cloud comes in front of the moon, there's only the starlight, so it has to diminish the light, otherwise there would be a light going on the sand. So even that works. In the other side of the squid, there are sensors which calculate the amount of light needed, and down there are the lamps. What are the lamps? They are small crypts of bacteria that produce light. The bacteria get a free lunch, like we got today, and the bacteria like to be there and produce the light, and they profit from it, and the squid profit. And it opens and closes the crypt to let more light or less light come out. And it's a fantastic system. I think the nature is full of such incredible things. I think the creator is a very nice person, and he has a good sense of humor because he's producing all these things for us to admire. And I tell about these 40 different animals in my book, The Animals Teach You. But this evidence is pretty interesting, because now that they found this soft tissue in a dinosaur, they have written that maybe in 50% of the dinosaur bones that they dig up all the time, they can find soft tissue, now that they started to look for it. Very interesting. Well, we talk about this a little bit, somebody asked how long have we been here. We talk about that nobody can have children without DNA copying mistakes, and these mistakes happen in every generation, and these mistakes take us toward dying out or extinction. When we look at the copy errors, we can see that human DNA has been at its best about 6,000 years ago. This goes for human intellect also. We're getting more stupid all the time, unfortunately. When we estimate the increasing errors in the future, it's evident that we will die out after a few thousand, not millions of years ago from now. Evolution claims that we have been here 100 or 200,000 years. That's not possible, because we would have died out 10 times because of the failing catastrophe, as the German says, mutational catastrophe. Naturalistic models of the exactness of copying do not explain these facts because they lead rapidly to an error catastrophe or mutational meltdown, as it said. Only the intelligent design model, where the original DNA is considered perfect and there has been a programmed error correction mechanism in operation during DNA replication, is in harmony with these study results. During the first human generation, there were no copying errors. Up to Abraham, only a few, and modern humans have about 4,000 copying errors. I mean copying errors, not mutations. There's lots more mutations. In reality, however, humans have today about 3 million mutations coming from other sources like radon, cosmic radiation, and stuff. The naturalistic evolution theory of life in a mutagenic world like ours for millions of years is not at all in harmony with these facts. Time is the enemy, because the more time, the more certain that everything will die out. In the created world before the fall, no errors happened, and Adam and Eve's children received perfect DNA. That's why Adam's and Eve's children could marry without no problem. God, who knows everything, knew that man would fall. He equipped the DNA information code with redundancy, which means extra information to tolerate the coming mutations. So he knew about it, he took it into consideration. And this is the biggest mystery in natural science today that we went through in the last lecture, is the origin of the DNA code. The genetic researcher Dr. John Sanford points in his book, The Genetic Entropy and the Mystery of the Genome, that human DNA is degrading and genetic entropy cannot be avoided. This is because of the fact that natural selection deletes only the most dangerous mutation. It can't see the hidden recessive mutations. I recommend Dr. Sanford's book, Genetic Entropy and the Mystery of the Genome. It was one time available free on the net. I don't know if it's there still, but this is a quick question. You ask, how long have we been here? This answers your question because we can see it from the mutations that we were perfect about 6,000 years ago. Then mutations started to accumulate, and now we're here. Most mutations are either neutral, that's because of the redundancy in the code, as far as we know, or only slightly harmful or have no effect at all. In this way, mutations can move from generation to generation and they accumulate in the human genome continually. Humankind's genome goes down about 1.5% every generation. Down, 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 down. There is a mathematical model in Sanford's book that shows that humans will die out in 300 generations or in 6,000 years. Different models, you can do different mathematics, show that the extinction time for us is from 1,000 years to 1.5 million years, the average being about 30,000 years, but not hundreds of millions of years or billions of years. Naturalists have no convincing model to deny these facts. This is a fatal blow to their claim that life has continued for millions of years. But the facts of genes from this study fit well with biblical creation and the fall that started this degeneration, or as we call it, genetic entropy. The all-knowing God foresaw that the fall and equipped his creation with many, many DNA repair systems. If God had not put DNA repair systems into the system, nothing would function here. This makes it possible that people today can still live a relatively healthy life, and it's possible for them to have healthy children for yet many generations in the future. Without these inbuilt repair mechanisms, life would have ended quite fast because of copying errors. This is called the error catastrophe. You see, the DNA replication you're all familiar with. I don't have to go into this. It's an extremely complicated thing. It copies letter by letter like 1,000 Bibles per hour, these molecular machines. 1,000 Bibles per hour, letter by letter. Makes very few mistakes. And this is the correcting mechanism which follows the replication. It's like a scanner. After replication, it scans the result and repairs immediately the mistakes. Very interesting, because without this repair mechanism, whatever you think about the original life, if you think the original life was some kind of RNA world with an RNA molecule that has enzymatic properties, it would need to copy itself only 10 times. All information would be lost without this correcting mechanism. We have to take out these details, that who put the correcting scanner and repair mechanism, which is very complicated, into the DNA to make life continue. How did it evolve? What happened before this correcting mechanism evolved? All information was lost. These kind of things we have to take into discussion. Well, I think we need soon some coffee because somebody is sleeping. That's okay. But also a relevant question is, why do cells go into so much trouble and spend huge resources? This costs money to prevent copying errors if mutations are claimed to be the engine of evolution. Let us ask this question. They do all they can to prevent mutations which are claimed to be the engine of evolution. Got to ask these questions. Depending on the copying and repair system, the quantity of errors seems to vary widely from 1 error per nucleotide to 1 error per 10 to 100 million copied nucleotides. The cells seem to have the ability to combine different systems and in cooperation reach a better result in repairing errors. However, on the whole, our cells are incapable to copy our genome's 3 billion nucleotides perfectly. Can't be done. So always going for the worst. Always going for the worst. In this fallen world, not getting better, everything is getting worse. Recently it was shown in a population study that the mutation frequency is about 1.2 to 100 million nucleotides in one generation. This means in the 3 billion nucleotide human genome, 36 single nucleotide changes per person and generation. Now this is important. The female egg cell that transmits life to the next generation is replicated only a very few times when compared to sperm that is produced 100 million cells a day. In addition, old men have many more mutations in their sperm than young ones, so don't marry an old man. After Kong, if the father is under 20, he transmits 20 new mutations to the child, but if he is 40s, he transmits 65. look at life's beginning. After creation, we have gone through about 250 generations. It's interesting because 250 generations seem to separate you and me from Adam and Eve. Quite fascinating. Every fertile woman produces only 24 generations of egg cells in the ovaries during the embryonic stage. This means that when a woman is born, there's 7 million eggs in the ovaries. Out of these young girls' eggs, only 1 or 2 million are finally selected to the mature ovaries. New eggs are probably not formed after that. The ladies are saving us because they produce very few mutations. And it's because of the ladies we exist here, because when Adam saw Eve for the first time, why did Adam call Eve Eve? Eve is Hebrew, and Eve means the mother of all living, because it's in the egg cell, which is huge, that is the mother of all living. We all inherit our cell organelles from our mother only. The father gives just the I don't know what to say. The father gives just the bad chromosomes, that's all. But the women are the key in survival. Women have all the information. And we can see from the genes that everybody has had the same mother. It's an interesting thing. But interesting thing is also that humans have gone through a bottleneck, which means that we have been many million, then there has been a catastrophe, and we descend from 8 people. That's a Noah's flood. Millions were killed, and humans have gone through a bottleneck, and that's why we are like brothers and sisters, be it black, yellow, whatever. We are so close to each other. That's a problem for evolution, because we're like one family. So the Bible is right. The Bible says that Eve is the mother of all living. Now we know it's true. Eve is the mother, woman is the mother of all living. Men, on the contrary, produce sperm through their lifetime, and they accumulate mutations. As I said before, 85% of the bad stuff comes from the father, only 15% from the mother, because they don't divide so much. The women rule. The role of female DNA is very important in maintaining life, because the egg, DNA and cytoplasm with all its organelles forms every person's first cell. Male gives only the chromosome. We inherit our cells directly from Eve, the mother of all living, Genesis 3:20, not from Adam. This we know to be true, and the Bible text has been shown to be correct now 6,000 years later. The importance of this will become clear later. Human population of today has gone through 23 times 250, 5,750 egg cell generations. When we look into the past, in the light of these studies, we can predict that the quality of the genome has been better in the past, and less DNA copying errors have taken place. When parents have children today, they transmit their millions of one nucleotide mutations with 36 new errors. If one of the parents has received, for instance, a Chernobyl high dose of radioactive radiation, he or she transmits more mutations. So, let's see. The ID model explains this very well. The naturalistic model does not. The third important, you can see the slides I went over because to save time, you can see them in the internet site of the society. And the third important consideration for this model is the fact that copying fidelity must become worse with an accelerating speed, because the copying system also accumulates more and more errors from one generation to another. This leads to a vicious circle. The copying system accumulates mutations, more and more errors, but the repair system cannot help because it gets errors also. So it's a vicious circle. It's more fast all the time, getting more sickness. So God must have designed a special way to protect the copying system from errors. The ID model works very well here. After the fall, it had an exactness of copying where one error happened in 23,000 billion nucleotides and cell generation for the system to fit in today's situation, which is one error per 2 billion nucleotides. Now I'll hop over this. When we talk about the fall, there man was deemed to die a death which presupposes sickness and becoming older. We know with Dr. Elliot that we are getting old and we are getting sick. But that's what the Bible means, because the death was introduced at the fall, because if there was no death, this misery that we're experiencing here would continue forever. But we're looking for the new heavens and the new earth where there will be no sickness. It's okay to get older and to get sicker. Paul talks about this in Romans chapter 8, where he says that, Paul says actually there the whole creation is subjected to decay, in other words, biological entropy. This is also biblical. It is quite clear that the naturalistic model does not fit in the real gene data, because it leads to error catastrophe so fast. The ID model fits well. Well, I hop over this. These basic starting points fit very well with the Bible's description of a perfect creation and a subsequent fall that took place some thousands of years ago. But they are impossible to fit into the naturalistic starting point. The naturalistic claim, the atheists claim, that life started with molecules that made copies of themselves without repair systems can now be seen as a failed fantasy. The RNA model fails so fast because of error catastrophe. We've been through that already. That is quite astonishing that this kind of pseudoscience can be taught in schools and universities as a believable origin of life model. You will be taught in the university that life started in an RNA world. But this doesn't work, because now you understand that if the RNA makes copies of itself without a proofreading and a copy repair system, information is lost very fast. But they don't go so far into the detail. Today we know that we are not related to other animals. Humans have the orphan genes, about 20% of genes, that our presumed animal forefathers do not have. And the RNA world model does not work because it has no copy error repair system. I want to underline this. And there are also, of course, many biochemical reasons. Because the RNA molecule is very fragile, and it is unclear where sufficient quantities of D ribose would come from. That is a special biochemical thing. You can read the original article in the Journal of Creation. I put this site creation.com, but there's more technical and academic journal, Journal of Creation 28 2014, and this will come maybe on the internet about Christmas time, so you can read the whole article. But go to the internet and look for creation.com or Journal of Creation, and you can get very good articles on this which give you information so you can defend the faith. We're working hard in Finland. With friends, we produce a journal in Finnish. This comes out about 3 times a year. It's got the color pictures and text in Finnish. But the same journal you can get in English, Creation Journal. It's very good because all the articles are made by philosophy doctors or doctor or something. And the articles are good. And you find always new things on this stuff. And as I said, I got some material. This is in Finnish, but it's a myth about 8 men. But this book is also can be found in English. It's called Goals of Contention. So I got here, which appeared a month ago, a book on the bacterial motor and the mechanism of evolution, or the lacking mechanism of evolution. I got some copies here. This is quite new, so this will cost a little more, 15 euros. But this is strong stuff. If an atheist reads this and still wants to be an atheist, so I think there's something else wrong. But let this be polite. I wrote a book on a TV series. Maybe you have a TV7 in Estonia also. So I had a series of there in 28 series about. But what I want to warmly recommend to you is this 1.5 hour movie in English on God of Wonders. This is very good. I mean, excellent. So you can get this for. If you want to take only this, you can get this for 3 euros. But if you want my book, you can get this book for 5 euros. But if you like animals, but this is not sort of in Finnish, you can have the animal book, the image of drawing book, and the film, all 3 of them for 10 euros. So you can take a look at that. So we do a lot of stuff, and there's lots of interest in Finland on this topic because I go lecture maybe 2-3 times a month somewhere in Finland on this topic, so people are very interested in it. But now it's the time to be critical and continue, because the problem is that when you think about the naturalism, it's like an island, and the truth is here. But here is there's a fence, like the Berlin Wall. Here is a fence, and here is the answer. Scientists come to this island to try and find the answer. They dig here, 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 they dig here. But they are not allowed to go on the other side of the wall. So this is why they will never find the answer, because to do science without considering a designer is not acceptable. Because I would say that is no science. In science, all possibilities should be considered. If your starting point is that there is no creator, so you work here, you not go here. But the answer is here, the designer is here. And that is a problem, because I would say that this evolution business is just a faith system. It's a religion, it's not science. Because once you say that our hypothesis, our theory is true, no complaints, just at that moment it stops being science. Because science, you have to be open for this also. And seeing the bacterial engine, you must understand that it's quite easy to show that there's design in nature, design all over the place.